Now we are going to discuss about DNA computing concepts for medical applications. Then we are going to discuss about overview of our project. The main objective of our project is to identify what type of cancer is occurred in particular patient by using different machine learning algorithms. So machine learning approaches are powerful techniques commonly employed for developing cancer prediction models using associated gene expression and mutation data. This manuscript provides a comprehensive review of recent cancer studies that have employed gene expression data from several cancer types for survival prediction, tumor identification, and stratification. So we also developed an overview of biomarker studies that are associated with these cancer types. The system is developed the different machine learning algorithms for predicting or classifying the type of breast cancer. Then finally, we can estimate some performance metrics such as accuracy, precision, recall, F1 score, ROC curve, and confusion matrix. Then we can compare the results based on accuracy in the form of graph. Then we are going to discuss about flow of our project. First, we need to take one input data gene breast cancer data set. The collected data we need to read and load our collected data with the help of pandas package. Then the collected data is carried out to pre-processing model. So in this step we can check or handle any missing values is present or not. Why we are checking missing values means for avoid wrong prediction. Then label encoding. So we need to convert the string into numeric integer value. Then pre-processed data is carried out to data splitting model. In this step, we can split the whole data into test and train. Test is used for prediction and train is used for evaluation. The splitted data is carried out to classification model. In this step, we are implemented the two different ML algorithms such as KNN and logistic regression. Then finally, so prediction. So with the help of classification algorithm, we can predict the type of breast cancer. Then finally, performance estimation. So we can find some performance such as accuracy, precision, recall, and F1 score. So finally, we can visualize your data in the form of graph. So this is the flow of our project. Then we are going to discuss about demo of our project. We are run our project in Spider IDE from Anaconda Navigator. This is the data selection. So here we can load our input data by using pandas package. Then pre-processing. So in pre-processing we can check any missing values is present or not. 0 indicates there is no missing values. So the 22 indicates that 22 missing values is present in corresponding columns. Then after checking missing values, so all the columns is considered as 0. So that means so we are removed the missing values or replace the missing values by 0. Then before label encoding, so these are the strings which is present in our data set. So after label encoding, we need to convert these string into numeric integer values. Then data splitting. So data splitting means we need to convert the test and train. So this is the total number of data and this is the total number of training data and testing data. Then this is the result for KNN. Accuracy KNN got accuracy 93% and classification report for KNN and confusion matrix for KNN. Then logistic regression. So A logistic regression got 90% accuracy and classification report, and this is the confusion matrix. So finally prediction for we are testing the some test data. So here we have taken a sample of 10 test data, so it will be considered like a type is breast cancer. Then finally comparison, when compared with KNN and logistic regression, so we conclude KNN is efficient for our project. So we can compare the result in the form of graph. Thank you.